So, imagine an industry where like 90% of everything you build over a decade just completely fails.
And somehow you still stay in business.
Right. Exactly. And that is unfortunately the reality of modern pharmaceuticals. 10 to 15 years of research, billions of dollars spent, and then 9 times out of 10, the drug just fails in clinical trials.
That's staggering.
It really is. But today we're looking at a stack of sources that suggest, well, a simple text prompt could drop that failure rate to near zero.
Which, I mean, that's a massive claim.
Yeah.
We are basically looking at a fundamental shift from, human trial and error to fully autonomous systems.
Yeah, the sources we have today are dense and honestly just paradigm shifting. We're pulling from this landmark perspective published in ACS Central Science.
Right, by researchers from In Silico Medicine and Lilly.
Yep. And we also have these technical reports on incredibly powerful open source biomolecular models. Specifically, we're looking at ByteDance's Protenyx V1.
And just to keep our feet planted firmly on the ground, we also brought in some critical forensic analyses detailing what industry insiders are calling the pharmaceutical superintelligence deficit.
Which is such an essential counterbalance, I think. It gives us this comprehensive look at both the staggering potential of the tech and, well, the very real physical hurdles that are still tripping it up.
Exactly.
So the mission for this deep dive is to explore the dawn of this pharmaceutical superintelligence, or PSI, as it's often called. We want to unpack the entire end-to-end prompt-to-drug pipeline.
Right. The idea of just typing a sentence and getting a medicine.
Yeah. Imagine sitting down and typing like, design a drug for idiopathic pulmonary fibrosis, which is a devastating disease where the lungs inexplicably scar over. You type that, hit enter, and the AI just goes to work.
It's wild to think about.
It really is. We're going to figure out how open source AI is democratizing all of this while also, taking a hard look at the reality check hurdles. Okay, let's unpack this.
So to really understand the magnitude of this shift, you have to look at how we've been doing this for the last century. Historically, discovering a new drug is it's highly fragmented.
Very siloed.
Extremely. You have biologists over here looking for a target in the body, then chemists in another building trying to build a molecule that affects that target.
And then clinical researchers figuring out how to actually test it on humans.
Exactly. It relies heavily on human intuition and isolated teams.
And I guess that siloed approach is exactly what leads to that 90% clinical failure rate.
Yeah, it's financially and scientifically exhausting. But what this ACS Central Science paper outlines is a structural transformation. They are moving toward this new paradigm defined by scientific intelligence.
Okay, scientific intelligence.
Right, which relies on a centralized orchestration layer.
The prompt-to-drug workflow. This is where it gets a little wild for me, to be honest. The idea is that at the top of this whole hierarchy, you have a central large language model, an LLM, acting as the intelligence layer.
You type in your prompt, and it manages the rest. But I have to push back here a little bit, because we've all seen generative AI do some completely bizarre things, like language models are notorious for hallucinating facts.
Oh, absolutely.
If I ask a chatbot to write a biography, it might invent a college I never even went to. So if we let an AI invent a drug from scratch, how do we prevent it from just, hallucinating a physically impossible molecule?
That is the most critical question right there. And it's the exact problem that multi-agent stratification solves.
Multi-agent stratification.
Basically, if that central language model tried to generate the chemical structure itself, it would absolutely hallucinate impossible molecules.
Because it just predicts the next word, doesn't know chemistry.
Exactly. It knows language, not physics.
Right.
So it doesn't actually create the drug.
It just acts as the boss.
Highly capable manager, yeah. It takes your plain language prompt and it breaks it down into discrete, highly technical tasks. Then it delegates the actual scientific work to specialized, biophysically grounded agent cohorts. Oh, okay.
So you have a biology cohort, a chemistry cohort, and a clinical cohort. Every single decision the language model makes is checked, validated, and executed by these specialized tools.
Tools that are strictly bound by the laws of physics and biology.
Precisely.
So it's not one giant brain just making things up. It's an orchestrator managing a team of hyper-specialized digital experts.
And those specialized experts are the ones doing the heavy lifting step by step.
Which I guess brings up the very first step. Because before you can invent a new chemical, you have to know what you are attacking.
You have to have a target.
Right. If you design the perfect, most potent molecule in the world, but it targets a biological mechanism that has nothing to do with the actual disease,
Then it's useless.
yeah, it's like writing flawless, bug-free code for an operating system that no one uses anymore.
That is a really great way to frame it. Failures in target selection incur the highest downstream clinical costs, by far. You spend a decade building a drug, it works perfectly on the protein you aimed it at, but the patient doesn't get better because, well, you aimed at the wrong protein.
So how does the AI solve that? Like, how does it find the right target for, say, that lung scarring disease I mentioned earlier?
Well, this is where the biology cohort steps in, and it's anchored by a system called Pandiomics Target Identification Pro.
Or TID Pro for short.
Right, TID Pro. The strategy here is essentially text in, target out, TITO. And it tackles this using what computer scientists call a positive unlabeled machine learning framework.
Okay, I'm going to need you to break that down. What does positive unlabeled mean when we're talking about human biology?
It's basically a clever way of handling incomplete data. In biology, we know certain biological targets definitely work because we already have approved drugs attached to them.
Okay, so those are the known positives.
Exactly. But for the vast majority of the human genome, we simply aren't sure yet. So instead of labeling those unknown proteins as negatives or failures, the system treats them as unlabeled candidates.
Ah, so it keeps an open mind about basically every protein in the body.
Right. It doesn't write anything off prematurely. Then TID Pro analyzes 22 distinct feature scores across all of these candidates, and 12 of those scores are based on heavy omics data.
Omics data meaning things like genomics and proteomics.
Yes, exactly.
It's basically the massive digital blueprints of our DNA and the proteins they create.
Exactly, those blueprints. It looks at the raw biological data. But then it pairs that with 10 feature scores based entirely on text-driven metrics from scientific literature.
So it's reading the raw biological code of the human body while simultaneously reading like the entire history of published medical research and cross-referencing them.
That's the text-in part of the strategy.
I saw a crazy statistic in the sources about how this compares to standard AI models. When tasked with retrieving clinical targets, how big is the gap between a generalized AI and this specific tool?
The gap is monumental. Standard frontier language models like GPT-4o, for instance, they hit somewhere between a 15 and 40% success rate when trying to find a valid clinical target.
Okay.
TID Pro achieves a 71.6% success rate.
Wait, really? That high?
Yeah, it completely crushes generalized models because it's purpose-built to merge language processing with hard biological data.
But finding a statistical correlation isn't enough, right? I mean, just because a protein shows up a lot in patients with lung scarring doesn't mean the protein is actually causing the lung scarring.
Right. Correlation is not causation,
It could just be an innocent bystander.
which is why the system has to prove its work.
Yeah.
And this is where another tool within the biology cohort comes into play, called BioReason Pro. It provides the mechanistic justification.
It explains the why.
Exactly. It actually generates structured reasoning traces. It deduces a protein's molecular function by looking at its domain architecture.
The specific physical building blocks that make up the protein.
Right. And it generates a logical narrative, proving that the protein plays a causal role in the pathophysiology of the disease. It ensures the AI suite is prioritizing targets that actually drive the illness.
Preventing the system from basically wasting millions of dollars chasing a phantom.
Exactly.
Okay, so the biology cohort has successfully identified the rogue protein causing the disease, and it has proven it mathematically and biologically. But identifying the target doesn't cure the disease.
No, it just gives you the coordinates.
Right, because a protein is a physical three-dimensional object in the body. You can't design a drug to attack it until you know exactly what that protein looks like in 3 dimensions. And historically, mapping the shape of a protein was incredibly slow, painstaking work.
It was the major bottleneck. I mean, the foundational molecular layer of rational drug design relies entirely on predicting these precise 3D structures and understanding how potential drugs might interact with them.
And for a long time, the state-of-the-art in this space was completely locked behind proprietary, closed-source models created by massive tech companies.
Yes, entirely black-boxed.
But the sources detail a massive breakthrough that seems to be democratizing this entire process. Protenix V1. The developers claim this is the first fully open-source model to match or even exceed the performance of closed-source giants like AlphaFold 3.
Which is a huge deal.
But, I mean, any developer can claim their model is the best. How do they actually prove that?
They proved it by leveling the playing field. The developers restricted Protenix V1 to the exact same training data cutoff as AlphaFold 3.
Meaning it couldn't learn from any data published after September 2021.
Exactly. They also restricted it to the exact same inference budgets, meaning they gave it the exact same amount of computational power to solve the problem.
Wow, okay.
By strictly adhering to those controlled conditions, they proved that the performance gap between open source models and proprietary giants isn't fundamental.
It proves that the open source community can build high-fidelity structural predictors just as well as the multi-billion-dollar closed labs.
Yes.
But here's where it gets really interesting. In the Protenix V1 report, they detail a feature called inference time scaling. The sources describe it almost like a literal control knob for accuracy. How does that mechanism actually work?
It's a brilliant shift in how we interact with these models. In traditional setups, you run a prompt, the model calculates an answer, and you get what you get.
Right.
But with inference time scaling, you can explicitly trade computational cost for higher accuracy.
So you're giving the model a larger budget to think, basically. But what is it actually doing with that extra time?
It is sampling different structural possibilities. Think of it like asking an architect to design a complex building. If you give them 1 hour, they will give you their best single guess at a blueprint.
Makes sense.
But if you give them a team of 100 architects and a week to work, they can draw up 100 different blueprints, compare them all, find the flaws in each, and present the absolute best version.
So the AI is exploring different parallel universes of protein folding and scoring them against each other.
That's a really good way to put it. It uses random seeds to explore. And the sources provide a striking example of why this matters.
That's right.
Antibody-antigen complexes are notoriously difficult to predict in 3 dimensions. If you run Protenix V1 with just one random seed, so one architect,
Yeah.
it has a success rate of about 36%. But if you turn that control knob up and give it a budget of 80 random seeds, the success rate jumps to nearly 48%.
Oh, wow. That's a massive jump.
It delivers a consistent improvement just by throwing more compute at the problem.
And the fact that models like Protenix V1, along with others mentioned in the sources like Boltz2 and Chai1, the fact that they're open source, that changes the whole ecosystem, doesn't it?
It changes everything. Proprietary engines like Isomorphic Labs, ISODDE, for example, they often operate as black boxes. You put your data in, you get an answer out, but you can't see the internal gears turning.
Right. You can't tweak the underlying code to fit your specific lab's needs.
Exactly. Open source models provide transparent tooling.
So the global scientific community can take these structural predictors and plug them seamlessly into their own autonomous agent workflows. There's no vendor lock-in.
Biology just becomes a software-shaped computational pipeline. Anyone with the compute power can string these tools together.
Which brings us to the most science fiction part of this entire deep dive.
Here we go.
We have the target identified. We've mapped its 3D structure flawlessly using these open source engines. Now the AI suite is ready to actually invent the drug. The orchestrator hands the blueprints over to the generative core. We are moving from summarizing biology to generating novel chemistry.
This is where open source structural models act as the essential engines for those high-level reasoning agents. They use atomistic binder generation methods to design the drug.
Designing the drug atom by atom. The sources describe the AI looking for these cryptic pockets on the surface of the protein. And it makes me think of an automated high-speed game of Tetris.
That's a great analogy.
But the AI isn't just dropping existing blocks, right? It's rapidly generating entirely new custom blocks, atom by atom, that perfectly fit into the target's unique crevices.
And it knows how to build those blocks because it was trained on massive synthetic data sets, like Teddy Seeker, which has 3.5 million clusters. The AI has analyzed millions of artificial biophysical interactions, giving it this deep, deep understanding of how molecules fit together.
It's incredible.
It builds the molecule atom by atom, constantly validating it against biophysical reward functions to ensure it isn't just a pretty picture on a screen, but a stable chemical entity. It uses tools like Proteina Complexa to design these chemical binders.
But the pipeline doesn't stop at the computer screen, and this is the part that just blows my mind. The digital blueprints for these newly invented molecules are handed off to the execution layer.
Right.
The digital orchestrator contacts agents like CPR agents and chem agents that manage automated laboratories.
These agents calculate the synthesis pathways, which are the actual chemical steps needed to create the molecule in the real world. Then they command robotic self-driving laboratories to literally mix the chemicals, apply the heat, and synthesize the physical compounds.
From a plain text prompt to an AI manager to a digital architect to a physical robot building the drug, it is spectacular.
It sounds like pure magic.
It really does. So we have this incredible, almost flawless success in the digital realm, but the sources are quick to throw a massive wet blanket on this when we look at the reality of the physical world.
We really have to look at the pharmaceutical superintelligence deficit. While the vision of fully autonomous drug design is aggressively backed by major pharmaceutical innovators, critical forensic analysis points out some massive blind spots. And we need to be clear, we're just reporting what the analysts have found here.
Absolutely, we're impartial on this. But the deficit report drops a heavy reality check. Out of 22 grand challenges in predictive modeling for drug discovery, exactly 14 remain completely or largely unaddressed by this current PSI framework.
14 out of 22.
Yeah. It creates what the analysts call a death valley between generating a potent binder on a computer and realizing a safe human medicine.
A major part of that death valley is the potency trap. The current AI suite is absolutely obsessed with binding affinity, which is how tightly the custom drug sticks to the target protein.
But binding affinity isn't everything.
No. In human physiology, a drug with perfect affinity is entirely useless if it's highly toxic, or, if the liver recognizes it as a foreign substance and clears it from the bloodstream in 5 minutes before it can even reach the lungs to do its job.
Oh, wow. Yeah.
The sources specifically warned about the danger of HERG blockade.
Wait, what is HERG blockade?
The HERG channel regulates the electrical system of the human heart. If the AI designs a drug that incidentally blocks that channel, it can cause lethal heart arrhythmias.
Oh, geez.
It's a catastrophic failure, no matter how perfectly the drug binds to the disease target. The AI doesn't always know to look out for that because it's so focused on the primary target.
Right. It's got tunnel vision. And there's another massive hurdle they mention, the manufacturability bottleneck. Let's say the AI designs a brilliant, safe molecule. When you try to turn it into a physical, solid pill that can sit on a pharmacy shelf, you run into polymorphism.
Which is how a molecule organizes itself in a solid state.
Right. It might look great on a screen or even suspended in a liquid in a lab, but when you try to crystallize it into a powder to press into a pill, it falls apart.
Or it turns into a useless sludge that the human stomach can't absorb.
Exactly. The analysts put it perfectly, I think. They said the AI is providing a brilliant blueprint, but the recipe for mass manufacturing human medicine is missing.
But the sources are clear that this isn't a dead end. It's just the next frontier for development. To bridge this death valley, researchers are working to integrate a pharmacology agent into the workflow.
Which would do what, exactly? Try to simulate the human body.
Precisely. They are using physiologically based pharmacokinetic models, or PBPK models, and tools like Metatrans and SmartSip.
Okay.
Instead of just looking at the protein target, these systems essentially build a digital liver, or a simulated human bloodstream. They run the AI's proposed drug through this simulation to see if specific enzymes break it down too fast or if it turns toxic before anyone actually commands a robot to synthesize it.
So it's forcing the AI to consider the messy, dynamic reality of human biology rather than just the sterile math of molecular binding.
Exactly. The technology is in its adolescence. It has mastered the theoretical physics and chemistry of the molecule, but it is still learning how to navigate a living system and the realities of industrial manufacturing.
So what does this all mean? We started with a simple prompt, design a drug. We've explored how target identification tools like TID Pro read massive amounts of literature and biological data to find the root cause of a disease.
We've seen how open source engines like Protenix V1 use inference time scaling to flawlessly map the 3D shapes of those targets.
Yeah. We discussed how generative models act like master architects, inventing novel molecules atom by atom to fit those shapes. And finally, how those digital blueprints are sent to robotic labs for physical synthesis.
It's a pipeline that could potentially slash development times from decades down to a matter of weeks, provided we can close those critical gaps in pharmacology and manufacturing. The integration of digital bloodstreams and dynamic modeling is the necessary next step to make this truly autonomous.
So for you listening, the next time you hear a news report about a major medical breakthrough, there is a rapidly growing chance that it wasn't discovered by a scientist staring through a microscope for 5 years. It might have been orchestrated by a multi-agent AI suite, autonomously exploring chemical space, balancing toxicological data, and directing robotic synthesis arms while you slept.
As operational efficiency gains approach 90% across the drug development lifecycle, we are nearing a horizon where the discovery phase is virtually instantaneous. This raises an important question for you to consider.
When computational design and robotic synthesis happen at the speed of thought, what happens to the future of medicine when the only bottleneck left is the inherent speed of human biology itself?